Good day, Homo sapiens. How are you? And welcome back to studying microbiology once again. And today we are covering the last topic of the first chapter, history and scope of microbiology. That's the chapter name, but we are going to study the future of microbiology. Future of microbiology. Guys, everyone would have thought their own future in one way or another way, but today we are going to study the future of microbiology, right? Guys, give me a second. I'm a little bit slow at the start, so I'm just checking if everything's working fine and we will be breezing through this topic. And just give me a second, I need to check if the live is working. Right. Let me just check this guy. Hurry up, don't circle. Let's see if it is lagging. Guys, you can skip forward if you want to. I mean, in the video. But to me, I'm just checking if it will. Guys, looks like my avatar is not working. Why are you not working? Alright, so we are closing this. I think it is working fine. If it doesn't work, it doesn't work. I meant the avatar. Oh, wait. I am sure. Right. Let's go to this. Let's go. Alright, guys. So this is the future of microbiology. Future, alright. By microbiology, we know that this is the study of microorganisms, and by future, that means that what will be leading to the after we study these microorganisms, right? Like, what is the end results of studying these microorganisms? What will we gain if we study these microorganisms, and what will happen if we don't study them? And to what extent can we study them? This is the future of microbiology, right? As the preceding section has shown, the microbiology has had a profound influence on the society. What of the future? They are saying that we have seen that microbiology had its impact in the past, like the plague disease, the AIDS, the bacillus anthracis, the tuberculosis, right? And it is influencing some kind of, harms or beneficials on the society these days in the present. But what about the future? Science writer Bernard Dixon is very optimistic about microbiology's future for two reasons. All right, so there is a scientist, Bernard Dixon, and he is very optimistic about microbiology's future. For what two reasons? First, microbiology has a clearer mission than do any other scientific discipline. All right, they are saying it has a clear mission. I guess it will be because we know that we are focusing on the microorganisms, like it is point focused on the microorganisms. I think maybe this is what the clear mission will be. Second, microbiology has great practical significance. Of course, the practicality goes along with microbiology, just like any other research discipline. When we study microorganisms, we have to do it practically. We can't theoretically say something about microorganisms unless we experiment on them, right? Second, Dixon notes that the microbiology is required both to face the threat of the new and re-emerging human infectious disease and to develop industrial technologies that are more efficient and environmental friendly. They are saying that Microbiology, guys, don't underestimate microbiology. I think people think that microbiology is of not that much importance, but the microbiology is required to face the human infectious disease, guys. Usually people call everything a disease, but infectious disease is a different thing. The infection or infectious diseases caused by microorganisms, and you won't believe how many infectious diseases we have. Actually, we have more infectious diseases than the normal diseases, right? Normal diseases is just a disorder in your body organs or in your body physiology or functions, right? Yet infections, they activate your immune system one way or another, right? And there is no other diseases than the infectious diseases that have wiped the human populations. And to develop the industrial technologies that are more efficient and environmentally friendly, guys, understand this term. If you see the technologies these days, it has more drawbacks, like more drawbacks in a long term than the beneficents. Okay, like look at the cars. It's producing the chlorofluorocarbons and these types of radicals, destroying our ozone layers and polluting our air. Look at the refrigerator. It's adding to the global temperatures, increasing the global temperatures. A lot of technologies, like some not some, most 99% I can say technologies, the chemical industries waste their chemicals into the water and into the soil, and making the soil and water unable to work normally at, like, it's such a sad story that what the industries are doing to our earth, and we know that, but we still are so stuck to the life of technologies. Like, we can think of a future without the technologies, most of the people, right? Like, we do want to live in the oceans, in the woods, and in the mountains, but try it. I dare you, try it, and you will know how much would you want to go back to the life that you had. So they are saying microbiology wants to develop the technologies that are environmentally friendly. The microorganisms are such monsters at doing great work without causing any damage to the environment. Guys, this is the true topic. Like producing antibiotics, unlike the chemically produced antibiotics, microorganisms which produce the antibiotics has no potential harm to the environment, because the microbes that produces are not harmful in any way, and it's actually more beneficial to the environment because it degrades the material and things around the air. Right. What are some of the most promising areas for the future microbiological research and their potential practical impacts? What are the promising areas, guys? Can you think of something? My mind goes directly to the genetic engineering, right? Like, this is the most promising areas, the genes, the DNA, which we were most unfamiliar with, and we had no knowledge or less knowledge on that part of the world, on the life. And when we know that part, the only field that, except the genetics field, right, that do the changing in the DNAs, we use microorganisms the most to manipulate the DNAs, right? So if we are able to manipulate the DNA to the extent that we can take their DNA and integrate into eukaryotic organisms, specifically these days yeast, microfungi or unicellular fungi that is used to produce eukaryotic proteins, right? So to my mind goes to that part of, there are still a lot of them. So they're saying, what is the most promising area in your thoughts for the future of microbiological research, right? And what will be their potential practical impacts? What kind of challenges do microbiologists face? This is all comes into the future, right? What area is most promising? What kind of challenges will the scientists face? A discussion of some aspects of the future of microbiology follows. So these some aspects are going to be explained. Medical microbiology. public health microbiology and immunology will continue to be areas of the intent insert research. Guys, do you notice one thing that is common, and that is the humanity? Medical microbiology, public health microbiology, and immunology all are related to the human health. Human comes first. That's just as it is. If we don't live, what are we going to do with the environment? So that's why the first priority in any field is humans. So that's why the medical, immunology, and the public health will always be the areas of intensive research, because we want to develop the technologies and that will benefit the humans, and we want to get rid of the organisms, or we want to get rid of the pollutants or anything that is not of any use to humans. New infectious diseases are continuously arising, and old diseases are once again becoming widespread and destructive. Because there were diseases, microorganisms like plague, you can say, AIDS, tuberculosis, and anthracis, like these kind of diseases, or you can say malaria as well. They destroyed the human population, but that was the part of the past. But no, these days it's reoccurring again because of their mutation. They are not gonna sit down and let the humans, or you can say the environment, destroy them. It's every organism's mechanism to mutate, to change, to adapt according to the environment they are facing. So it's because of our carelessness that the microorganisms are developing resistance, and they are changing themselves to be more destructive and to be more resistant to the antibiotics that we produce. And what kind of diseases are included in this? AIDS aids is a syndrome, right? Immunodeficiency syndrome. Wait, what does the A means again? Wait. I completely forgot. Stands for. Oh, acquired immunodeficiency syndrome. Right. So this is the advanced stage of HIV, the human immunodeficiency virus, right? And it has more diseases than one combined in an organism that's supposed to be healthy, but because of the attack on the T cells, they become unable to fight against other organisms, other pathogens, and they develop multiple diseases at once. And that's the reason that the patients with the AIDS die, not because of one infection, but because of multiple infections at once. The SARS is, if you remember, SARS-CoV-2. This is for the recent corona, but this is the complete class of it. And I forgot the abbreviation of it. Severe acute respiratory syndrome. That is the whole class, right? This is syndrome. This can be caused by multiple strains of bacteria, or not bacteria, but virus. And by SARS-associated coronavirus, right? Coronavirus is not the name of a single virus or a species name. It is the name of a group of species. You can say a class. So this is related to respiratory tract. This is related to the immune system. The hemorrhagic fevers, hemorrhagic, the blood obstruction happening in the fever. These are hemorrhagic fever, the symptoms of many infections, not just one. Tuberculosis, we all know that this infects the lungs as well. Microbiology, they are excellent examples of new and re-emerging infectious diseases. Right, guys, this AIDS is a disease that's been there for a whole, no, for years, I think. We can't just get rid of it. It's there. We only have diseases that can prolong the life of the patient, that is being infected with the AIDS, but we can't get rid of the virus. We can't get rid of the SARS virus. It's dormant infection in patients. The were the tuberculosis. Like once you get a tuberculosis, right? The patient may recover from the tuberculosis, but it will get a tuberculosis again if you let the virus have that environment once again. It might not be the same strain, it might be different strain, but the tuberculosis is not going anywhere once you get infected with it. So, and they are happening again, and not just one or two patients are coming out of these, infections. A lot of people. I think that will be causing plague. You call them plague pandemic. Like these kind of high level these infections they will be causing. Microbiologists will have to respond to these threats. And the one that can deal with them will be the microbiologist, of course, the ones that are more familiar with the microorganisms. Many of them presently unknown. Guys, so these are what we know. Just as coronavirus was a new emerging infection and it was so sudden, like we don't know what to do with it, and we didn't know. So it's possible that threats of these kinds can emerge again, and it won't be corona this time. It may be another one that's way more dangerous than the coronavirus. So they will also need to find ways to stop the spread of the establishment in infectious diseases, as well as the spread of multiple antibiotic resistance. Guys, the development of microbial resistant microorganism is one thing, right? Or you can say the recurrent infectious diseases are one thing, but the multiple antibiotic resistance, this is so dangerous. And these days everybody has microbes that are super super bugs. Yeah, they call it super bugs. There are microorganisms in your body that is resistant to every single type of antibiotic that is being produced. I think they're the bacillus, no, the Mycobacterium tuberculosis. Wait, where are the super bugs? Like there are a lot of microorganisms that are considered super bugs because they are resistant to everything and you can't get rid of them. So that's why we use multiple drug therapies for them. Like we can't use one antibiotic for them because they are resistant to it. And I think scientists are working, not I think, but scientists are working to get rid of these super bugs by one way or another way, but they are not succeeding very well in that. Yeah, methicillin-resistant Staphylococcus aureus, Clostridium difficile, vancomycin-resistant Enterococci, multi-drug-resistant Mycobacterium tuberculosis. Yeah, so these are all super bugs. All right, so this is not just the bacteria. This can be any microorganisms that have become resistant to antibiotics or other medicines, right? Present till day. So the first thing we said that we have to see the future of medical microbiology, and the future of medical biology will be that we have good that we there will come a time that we will have to deal with these multiple antibiotic resistance bacteria and the re-emergence of the infectious diseases and be more stronger than the previous attacks. The microbiologist will also be called upon to create new drugs and vaccines to study the association between infectious agents and the chronic disease, for example, autoimmune and cardiovascular diseases. Right. So the microbiologists do deal with the microorganisms, right? And they will have to wait. So it kind of stopped. I think the limit of the mic was done. everything's working. They also have to study the association between infectious agents and the chronic diseases, right? So the in microbiology we do study the autoimmune diseases because it is involved, with the autoimmune, with the immune system. Study the immune system because all microorganisms, maybe one or two exceptions, they cause the immune response in a body. So, and to further out understanding thorough understanding of the host defense and how pathogens interact with the host cell. It will be necessary to use techniques in a molecular and recombinant DNA technology to solve many of the problems. If your cell wall is being resistant, if your cell membrane is being resistant, if your ribosome is being resistant, I think the only way possible for us to get rid of some microorganisms will have to deal with the DNAs or RNAs. And I think the recombinant DNA technology have been popular since few decades, but the true development is still, that threshold still is not reached where we have full understanding of DNA, where we have full understanding of DNA technologies and DNA, how the DNA completely works, and the enzymes that cut it and the enzymes that combine it. Like, so we have to study that. There's still far way road for us to be on that level that we can manipulate DNA as we want. Industrial microbiology and environmental microbiology also face many challenges and opportunities, guys. After humans, the area that comes is the surrounding of microorganisms, that is environmental microbiology and industrial microbiology. Industrial is more like advanced, like it is not that much necessary, but to work on a higher level, on the larger scale, we do use industrial microbiology. And if you want to be advanced in anything, we have to go towards the industrial microbiology. Before that, the environmental microbiology. They are saying there are challenges and opportunities as well. We all know that the environment is becoming not so harmful for just the larger microorganisms, but it is becoming so hazardous for the microorganisms that can survive in any extreme conditions more likely. They are having trouble with growing and surviving in the environment. How do you suppose that we will live in that environment as well? So microbiologists are studying like what is the problem with the environment that they are causing that many problems to the microorganisms, or what are the microorganisms that causes the problems to our environment. So microorganisms are increasingly important in industry and environmental control. Guys, we can understand the industry part because in industries we use microorganisms, we multiply them and use them as slaves, or you can say laborers, to produce what we want. And we switch off the genes that are not necessary for their growth, for their survival. And other than that, we turn off all of the genes and let them produce only one thing that we need. So we do use them in higher numbers and higher amount. And yeah, environmental control. Guys, have you heard of the microorganisms that get rid of the plastics? So they are developing the microorganisms that can get rid of the plastics for us. The plastic that is the biggest problem in our environment, for quite some decades now. For example, microorganisms can serve as a source of high quality food and other practical products. So this is talking about the industrial microbiology, right? There are microorganisms like, you can say, the algae, and there is a fungi, corn protein, like that, we call it corn protein. Like, they have high number of, maybe some have high number of proteins, high amount of carbohydrates, and the two things that we need. And the fats, we don't usually use one for fat, like we don't need fats in our body as carbohydrates, and you can say proteins. So they are used as a source of food and other practical products such as enzymes for the industrial applications. They may also be used to degrade pollutants and toxic waste and as a vector to treat diseases and enhance agricultural productivity. Let me explain it one by one. to degrade pollutants. I have given example of the plastic, but plastic is higher difficulty pollutant. Like there, the microorganisms need to get used to the pollutant for it to degrade it. Are there microorganisms that just have the mechanism or enzymes in them that can degrade certain pollutants? They can degrade all of the pollutants, but there is a type of pollutant they can degrade, and everyone is different. Toxic waste, they do degrade that as well, and as a vector to treat diseases. So there are microorganisms. How can they be vectors to treat diseases, right? Microorganisms to treat vector disease. Guys, I think the best example will be the vaccines. Like we do use the killed or slightly killed microorganisms to cause the to treat the disease that they are causing, like vaccines. Vaccines, what kind of vaccines? Like corona vaccine, kill the coronavirus, or use some part of the coronavirus and inject it in our body. It causes our immune system to be triggered and produce antibodies, and in turn they treat the disease. But I think they don't mean that kind of vectors here. Let me search it. What do they mean by this? Viral vectors are used in gene therapy. All right, so they are talking about this one, gene therapy. Guys, viruses are the best source to make our DNA, or to make our wanting DNA to reach to the higher amount of cells in lower time. We do know that the virus is replicate, quite a number in a second, and it has only genes, or you can say DNA, and only proteins. The protein is just a covering, so all they have is DNA. So they replicate in at high rates, and they are small, so they can go through every cell, and they combine their own DNA with the host DNA. Some Most of the viruses do that mechanism, like combine their DNA to the host DNA, and when they are done with it, they destroy it, and they destroy the host DNA and leave their own. But there are some ones that don't destroy the host DNA, and we can use these viruses to to use them as a transporter to make them reach our DNA to the host that we wanna make it, and they are used as gene therapy. Like for example, there are genetic diseases that is caused by certain gene, and we want to get rid of that gene or replace that gene with the correct one. Like we can't do it mechanically, because how are we going to change the single genes of every cell, a cell that we can't even see? Well, our tip of the pen has millions of cells. How are we going to treat it mechanically, or with our eyes, or with our hands? So we use viruses that are so small, and they are efficient in reaching to the every cells, and they go there and replace the gene. They don't do it intentionally, right? This is just their mechanism to do their work, and we use that to our advantage, and this way we do the gene therapy. I think gene therapy is not used that much in the current world. Yeah, gene therapy is still not that advanced, because there is a high risk of changing your host DNA and making something, outrageous, or you can kill them. The development of techniques, especially DNA-based technologies that allows the study of microorganisms in their natural environment, has greatly stimulated research in the microbial ecology. So they are saying DNA-based techniques have allowed us to study the microorganisms in their natural environment. I don't know how they did that. How do you study the microorganisms through genes? Or are you saying that once we decode what does this mean? Guys, this needs to be asked from ChatGPT. Google doesn't answer things like that. go. What does it mean? The answer is so stupid. Do they think I don't understand what this is now? I'm asking how exactly. Oh, so it is the same thing. So, like, we don't have to go and see how the microorganisms act in the environment directly. We just need to get the sequence out, and we know what proteins they make, and from the DNA, right? Because all of this includes the DNA. We can study DNA, and in turn, we know how do they live within the environment. Or we can take their DNA and insert into another organisms, right? And like we don't have to directly see that organisms do its work. We can see the replaced microorganisms and see its work. Still high thorough sequencing next generation. This is just to read out the DNA. It is also the sequencing. This is also the sequencing. All of this is the sequencing. Where was I? Yeah, here. Several areas of the research will continue to be important. Understanding microbial diversity is one area that requires further research. Like there is no end to this area of the research. Every day passes and a new microorganism emerges. How are you going to understand this diversity? So this area just needs further research because there is just no end to it. It is estimated that less than 1% of the Earth's microbe have been cultured. 1%, are you kidding me? We break our brains to remember all of the microorganisms, and when we go to that site where there is a list of the classes, genus of all of these microorganisms, they are saying there is still this 1% that we have cultured. Well, they must mean that we haven't have the technology to culture all of the microorganisms that we have studied. But still, unless we culture them, we can't study them. So that means that only till now we have found out only 1% of the microorganisms in the environment. No wonder the microbial diversity requires further research. Greater efforts to grow previously uncultivated microbes will be required. Much work also, alright, there are some microorganisms that we don't culture and just go smash them under the microscope and we just study them. So they are saying we need to culture them as well. Much work also needs to be done on microorganisms living in extreme environments. Yeah, because first of all, to go into an extreme environment, we humans need more work for that. Like, how are you going to take out the microorganisms that in the lava, or you can say in the Antarctica or under high pressure? So to provide these kinds of environment in the lab is not for every lab. The discovery of the new and unusual microorganisms may well lead to the further advances in the development of the new antimicrobial agents, industrial processes, and bioremediations. New and unusual microorganisms. If we found out these unusual microorganisms, that will lead to the further advancement in new antimicrobial agents, right? We might get a hint from the microorganism, just as we got a hint from the penicillium notatum, or was it carcinogen, to produce antibiotic. We may get something else from the other microorganisms for the antimicrobial agents. We might have an idea what other processes we can do by using microbes, and bioremediation. Bioremediation is needed, so much needed. Bioremediation means to use the microorganism to remove the toxic compounds or the to make useful compounds from the present toxic compounds in the environment. Another area of increasing interest to the microbial ecologist is biofilms. Yeah, that's true. Guys, biofilms is the layer of the microbes in which they are attached cell to cell, and they do produce signals. They do send signals to one another to create that biofilm. Once that biofilm is formed, it is very difficult to get rid of the microorganisms from that area. Just think of your teeth. There is always a biofilm of the microorganisms on your teeth, and you just can't get rid of them no matter how much you brush it every day. By the way, we don't have to get rid of them because they are important for our mouth health. But still, the biofilms are just so intriguing to the scientists because they just can't get rid of them, and they have this strong mechanism. Unlike when they are in their singular forms, they are not that strength. They don't have that strength to live longer in the environment. But when they create biofilms, their ability to survive just goes tenfold or hundredfold. Microbes often form biofilms on the surfaces, and in doing so exhibit a physiology that differs from that observed when they live freely on the planktonically. Plankton is an algae, right? Algae? Am I kidding? Was that algae? Planktonically. Platonically. They did use the word plankton, right? Planktonically. Yeah, they did use the word plankton. The Google don't have any idea what can be the plank. Of course I know what the oh here it is. Superlative most planktonic. Floating or weakly swimming animal or plant life or life. Free living, standard weekly. Where did everything go? Right. So the way they live freely, it doesn't have to be on the surface. They can live suspendedly, right? Or weakly in water. For instance, microbes in biofilm are often more resistant to killing agents than they are when they are not in biofilm. Why? Biofilms are not only of interest to microbial ecologists, they can form on the human tissues as well. Just as I said, they have more degree of survival rate when they are in the biofilm stage rather than in the living stage. That's what that said, that they are more resistant to killing agents. And they are saying that they are interest of microbiology, microbial ecologists because they can form the biofilms on the human tissues or in the dwelling catheters. Catheters. It is a non-living thing. Dwelling catheters. A thin tube that is inserted into your bladder to drain urine. Why there? Or it's the same thing. Dwelling, it's not dwelling catheter, it's indwelling catheters. So it is a tube that they inject in a urine tube or what for the patients. So they can form biofilms there as well, and another main medical devices. All right, so this is not just a single example. They can they can form the biofilms in any medical devices, mostly which have the nutrients. In fact, microbial ecologists and medical microbiologists now understand that microorganisms are essential partners with higher organisms, right? They are saying they are partners with higher organisms. Of course, without microorganisms, the survival rate is, I don't think where we will be without microorganisms. Greater knowledge of the nature of the symbiotic relationship can help, guys. Of course, we won't be here on the earth if we didn't have microorganisms. The plants, which is the start of the food chain, they need microorganisms to grow. Without the microorganisms, they don't have any way to get that nitrogen from the environment. So that means if the plant can survive, we don't survive as well. I know we will be. We won't be here if we didn't have microorganisms. So they are saying if we found out or we find the knowledge of the symbiotic relationship of these microorganisms, it will improve our appreciation of the living world. Right? We will appreciate the living world more. We will appreciate the microorganisms more. It also will lead to a new approaches in the treating infectious diseases in the livestock and in humans. Right. The more we know something about something, we get more advanced in them, and we can, we get knowledgeable how to use them in multiple areas rather than one. The field of the genomics and proteomics have and will continue to have tremendous impact on the microbiology. Why is that? This is simple. DNA is the basic of all living cells. Microorganisms are tiny and are mostly unicellular. Well, they are multicellular, but they are mostly unicellular. And the unicellular cells have only one machinery that drives them to work, and that is DNA, that is their nucleus. Well, prokaryotes don't have a nucleus, they have suspended DNA. So the more we know about organisms, because that's what the cell revolves around it. Unlike the higher organisms, well, their DNA matters as well, but their functions are so difficult and complexly coordinated that just genomics, the higher organisms won't work that well unless you study their physiology and other characteristics as well. The genome of many microorganisms have already have been sequenced and many more will be determined in the coming years. Right? The more microorganisms we discover, the more genes we are going to discover. The more sequences we will get. These sequences are ideal. These sequences are used, the DNA sequences, how the one microorganism or how the one organism is related to another. Are they related in somehow ancient time? We know the ancient and the relation. Well, not me, but scientists, or you can say the ones that are involved in the genes. The sequences is ideal for learning how the genome is related to the cell structure and the function, and for providing insights into the fundamental question in the biology, such as how complex cellular structure develop and how cells communicate with one another and respond to the environment. Guys, unlike the body that have brain, cells don't have brain actually. Cells have DNA, but DNA don't work like brain. Don't think. And how do they communicate with one another and respond to the environment? How do they create such cellular structures complex? Like how when they are born and how they are made, how does they start working like they are professionals at that? They teach the cells to make the cellular structures as they are in the present. They do it on their own. Like how do they do that? The more we study about the genes, the more we will find out how these cells work, what leads them to communicate and what leads them to what leads them to be complex and what leads them to be simple. Analysis of the genome and its activity will require continuing advances in the field of bioinformatics. Guys, the information we get from the genes, first of all, genes are, you can say DNA is long and has multiple genes, not multiple, what am I saying? Millions and trillions of genes if you go towards the higher organism. How are you going to contain that amount of information in your brain? Humans can't do that. That's impossible. What if microwave or gene or alien arrives and they can store all of this information? From my perspective, average human cannot store all of this information. For that, bioinformatics is needed. If we can't even store them, how can we analyze them at all? So that's what bioinformatics and the tools come and they analyze the genome, they store the information. So the analysis of the gene and the advances in the genome, I think, relies on the bioinformatics. We need to have the bioinformatics information if you want to study